Okay, welcome back, everybody. It gives me great pleasure to introduce Dr. Eric Schadt, who many of you know is a director of the Institute for Genomics and Genetics, and he in turn will introduce our next keynote speaker. Eric?
Great, thank you, Scott. And so far, it's been an amazing lineup, and I think to complement what you've heard so far, it's a lot of what's driving innovation today in the medical sciences is of course the genomics and how we can use genomics to drive decision making in the clinical arena, and how we can push that even into the hands of consumers. And I think nothing epitomizes that movement more than what BGI is doing today. So it's really a great honor for me to be able to introduce Wang Jun, who heads, is the director of BGI, and has been like on this rocket ship for the last decade plus, starting with the sequencing in the early days of the human genome, where he literally raised his hand at one of those the early human genome meetings and committed China without, I believe, the knowledge of the government of China to contribute to that effort, and from there on just exploded into this amazing machine of tackling all the hardest genomic problems. So think, the rice genome and the silkworm genome and the pig genome and the diploid Asian human genome. Roughly, of the many, many papers Wang Jun and his groups have published, scores of those occurring in Science and Nature and on the covers of those prestigious journals, just again rocking the planet, going from zero to amazingness in a very short period of time, and I think really schooling a lot of what was happening here in the US of what was needed, and in particular taking a heavy informatics focus in building out an amazing informatics team to process, not just generate the data, but to process it. And so he's going to speak to us today on how they're actually driving the prices of that technology down to the point where it can go direct to consumer and have a very direct impact on human health. So with that, introduce Wang Jun.
Thank you, Eric, very much for the kind introduction. I'm here, actually, I'm really honored to be invited to give a sort of summary of what BGI is doing. I just try to make a very kind of comprehensive presentation, including almost all different kind of perspective of what we are doing, but it's going to be very fast. Well, life is digital. I understand people kind of mixed up with the digitalized information in computers and a human actually sitting or standing in front of you. But it's really true, the life is really digital. If you consider, for example, DNA, it's not just a form of kind of double helix, but it's actually storage. And we know 1.5 petagram sort of DNA contains 3 billion characters there, which is probably one of the most powerful storage in the planet. So imagine in the future, probably the library should be just stored in some sort of DNA on 96 wells. So it's really sort of cost saving, and it could really last more than 10,000 years, which cannot, any of those storage forms cannot really do that. But anyway, so the DNA is just actually a storage of life information, brain as computing, or probably every cell is a computing power there. So everything is really digital. That also sort of tells you, if you program ever for C language or any kind of those language, you need a model, right? You need actually a kind of functional model to say, this is really the starting point. And the gene act as a functional model of a language of life. So the gene could be very selfish in a way, right? So they try to expand themselves, copy here and there, but they actually sort of organize together in a different form as a genome. So different species take a different sort of surviving strategy in a way to combine different genes together. So in a way, a species may like something like vectors of genes that try to sort of expand themselves there. If you look at the evolutionary process, every different species, until now we understand, they all take different surviving strategy. And if you look at actually the tree of life in a way, the only feedback that to sort of test if the surviving strategy is true or not is to see if they could adapt to the environment or not. So this is actually a very nice feedback loop trying to sort of test the combination of genes and to see if it works. So we've already, to really understand the language of life, we started to sequence lots of plant animals, just like Eric just mentioned, rice, silkworm, anything. So of course, I mean, nature prefer those beautiful pictures as covers there. So we actually published lots of cover, a lot just the science, but also the beauty of the species there. But anyway, so we've sequenced many of them on the species level because we know those species kind of share some of those functional modules with human. And the Human Genome Project is really one of the great success previously for the first of the sort of reference or the life information or life language of human as a species there. But if you understand, you look into this species, not just talking about on the species level, but if you look at the individual level or the population level. So they're actually taking even different, subtle changes of surviving strategy when they're out of Africa there. So one of the important projects to sort of tackle that problems is the Southern Genomes Project. The Southern Genomes Project picked up all these sort of major populations worldwide and started to sequence them to see the population difference between each populations and to see how that actually affect their sort of adaptations to different environment there. One example, so it's not really in the Southern Genomes Project, but one of the examples that we've done for a Tibetan study shows a very interesting phenotype adaptation there. We all know the Tibetans living at a very high altitude there. And they actually could really adapt very well. So we sequenced a lot of Han Chinese and Tibetans and discovered this one gene called EPAS1 gene, which is really enriched in Tibetans, almost 90%, but very, very few in the Han Chinese, less than 10% of them. It's actually have a very high associated with the blood hemoglobin levels there and under a very positive selection. And recently we discovered this particular haplotype in the Tibetans actually inherited from Denisova genome, which is quite interesting. It got published in Nature a few months ago. But anyway, so that just tells you that different kind of combination of genes, changes in the genes, substitutions or haplotypes or any type of those different kind of genetic difference may have a big impact on kind of adaptation to the different environments there. Comes to the individual level, those we know, the population level stuff. We actually also did the Han Chinese, the Dai Chinese, which is the southern west population of China, also have different sort of genomes adaptations to different diets. But anyway, so you can think about all this population-specific stuff. But coming to the individual stuff, so we really want to know, what's your genome and actually what's in your genomes there. So we first sequenced the first Asian back to 4 years ago, or 5 or 6 years ago. The paper got published 4 years ago. The first information you can get is the history of yourself, right? So not just sequence the current DNA like the current population we can access to, but also all these kind of Asian DNAs, Asian populations, right? So we've sequenced the 4,000 years ago Schadt genome. We've sequenced the Aboriginal Australian genome. So we've sequenced a lot of those so-called Mami genomes. Well, so all these Asian DNA was still connecting them and trying to understand the history of certain sort of human migration paths there. But of course, you could also link it to your own genome, right? So to understand your own history, which is particularly very interesting for the country like China. It's a huge and heterogeneous populations there. And we all know, for example, for Han Chinese 6,000 years ago, 70% of Han Chinese actually came from from Y chromosome analysis, comes from 3 individuals, which we call Yan, Huang, and Zhiyou, which is basically we call ourselves as Yan Huang descendants. But anyway, so this is just to tell you, by sequence everybody, you can get a lot of histories of yourself there. Do we actually need to have more genomes to be sequenced? Is that really useful to sequence everybody's genome? Well, look at, look at the lifespan of any individuals there and different kind of status of if they're healthy or disease type. So comes to the first actually very definite answers that we need to sequence the genome is for the monogenetic trees, right? So people really got diagnosed as monogenetic traits. Sometimes the lifespan are very, very short there. So BGI initiated a so-called birth defects projects, right? So using non-invasive testing from maternal blood, we did Down syndrome testing, monogenic disorders, and actually, if you think about the rare in the population, but actually add them up, it's not that rare. So in China, it's about a few percent of the total population may have different monogenic forms of disorders there. So we've started a project locally, actually first to screen in the Shenzhen cities. So it's trying to screen every newborn baby for those kind of genome sequence to see if they have any monogenic disorders there, or even before that, actually. Well, with more and more this kind of consortium works or social networks of different individuals, those undiagnosed disease will be more and more sort of decoded, right? People will understand that. I just want to show one more sort of cases here for de novo mutation analysis of autism, just try to show you the personalized or the private mutation is actually very functional there. So we've collaborating with UCSD to finish up a kind of twin sequencing for the autism cases there. From that study, it's really a lot of people, sort of hospitals also, be part of these autism projects. But anyway, so first of all, you find out the parental ages there. So if parental age is 1 year older, you get actually one more de novo mutations there, which is clearly sort of make people like me worry. I want to have baby earlier in a way. But they are not that random distributed in the genome, so they are actually clustered together. If you look at those clusters, they have those hot spots or have the cold spots there. And if you zoom in there, they are actually, it's kind of interesting U-shape sort of turns out. So I was sort of having a hypothesis there, if the de novo mutation clustered into a region, those regions, if you compare human and chimp, they would be less conserved. But the results is opposite. They are actually more conserved in a way. So that tells you that kind of region are highly functional. So if you look at all those regions, they are sort of enriched by disease genes. And if you look at categories of different sort of disease genes there, they are brain development genes there. So again, it tells you all those de novo mutations happen in certain regions, and those regions are enriched by the brain development genes. But that just tries to show you those mutations, they're only very few, right? So every generation have something like 60 to 100 de novo mutations there. And those de novo mutation happens to be very, very functional. I mean, compared with those common variations in the human population there. So we can use that to do a lot of diagnosis, right? You can sequence a trail, then sort of to detect all the de novo mutations there and to see if they have any clinical relevance there. Well, this is one thing, right? But another thing is for actually, it's not just the monogenic disorders there. How about the cancer, for example? So the cancer is sort of, the cancer problem is different in a way because most of them are somatic mutations there. So to really understand the somatic mutation, you need to understand the cell lineage. So in order to do a cell lineage, you actually need to not just sequence very deep for tumor samples, but also try to do it in the single cell level. Can we actually get the single cell of cancer tissues and the normal tissues and sequence it in the single cell level? And the answer, of course, is yes, right? So we've developed all the single cell sequencing technology, and right now it could be done in a very efficient way. And this is just one example we've done for the bladder cancers there. By sequence every single cell, you could basically review back the tumor progression process, and you know exactly which gene had a somatic mutation that caused the separation between tumor and normal, and what kind of mutation happens because the subgroup of the tumors. But anyway, so those kind of single cell technology should be done in the future for every cancer patients, and we've started from a so-called cancer panel there to target on those druggable genes that could be used for every cancer patients there. So we've started collaboration in several hospitals in China, sort of try to sequence all of those tumor, whatever, panels there for every lung cancer, prostate cancers, and gastric cancer patients there in several hospitals. This is just one successful case that we've done almost 2.5 years ago for prostate cancer patients there. And actually, we've identified these very interesting pathways and also corresponding drugs there. But anyway, so this patient was lucky, so he got really treated afterwards. But those kind of study needs to be really expanded. Well, if you really do it right, of course, the curves could really make it a little better to try to make you have like a 5-year survival rate is a little bit higher there. But if you could really do it a little earlier, so you can do early diagnosis of cancer. CTC is one of them, right? The circulating tumor cell. So we started to screen a big number of so-called elder person, but actually without any phenotype of cancers or tumors. And we actually found out a kind of substantial percentage, like 1% of those screened persons have a very, very sort of important CTC somatic mutations there. Actually, after that, they go to do a normal sort of detection by ultrasound or whatever CT type. They actually discover something different. So this is just try to tell you, but still we're wrapping up the results there. But by actually a big screening of every individuals for the CTC will be a very sort of valid tools of early diagnosis of cancers there. Yeah, immune cell testing is another interesting stuff that try to see how diverse those immune cells are, which is, again, another way to tag into the cancer problems there. But people are more interested, I mean, of course, it's important for the birth defects, cancers, but people are more interested with chronic disease, which is a big problem. Type diabetes, hypertension, obesity, and all these issues there. We know the genetic risk variance there. They have actually very high or common in the variation in the population, but they probably have no impact. Or sometimes, as I mentioned to you, the rare categories of variations may have higher impact there. So people have done this GWAS analysis for many of those traits, right? So they've discovered a lot of interesting genetic risk loci there. But just to give you an example on the type diabetes, so even they are actually discovered almost 40 of those risk loci there. Add them up, it's only something like 10% or less than 10% of the heritability of type diabetes there. So people are talking about what's the missing part of that, right? So they have to go a little bit further. Is that as 1,000 type diabetes sequence is enough? Is about 100,000 type diabetes patient is enough? Or 1 million type diabetes patient sequence is enough? But I would say we need to sequence every type diabetes patients in a way. But anyway, so that just to move the curve from those so-called common variants to the rare shifting there. The ideal study in the future, I mean, people are talking about different projects. They all try to find a shortcut of somehow, using a cancer panel, or just study 1,000 genomes without a phenotype data, or sometimes just wanting to have one candidate genes, a single gene studies there. But eventually, you actually need to have 1 million genome with whole genome sequence with full clinical data. By that, you could study the traits, not just the disease traits, but the traits like heights, even IQ score. That's actually Steve Su just mentioned. You need 1 million sort of individuals to tag that traits. But anyway, so eventually the idea, if you sequence enough individuals with full clinical data or phenotype traits data, so the idea is try to have this formula, right? Based on all these omics in different environment, they have different phenotype. Then we miss one part, environment. Well, I mean, I'm not talking about air pollution in Beijing, which is clearly a problem there. Not in the APAC time, but now it's getting worse, probably. I'm not also talking about anything sort of chemical or anything sort of we cannot really control, or we can, but we didn't really do that. But anyway, so there is actually a biological environment that we should tag on, for example, virus, right? So this is just one example about HPV integration with the human genome for the liver cancer in China. But we also have the HPV problems. We also have all this kind of unknown virus, which we needed to do a large-scale discovery sort of efforts there, and also to understand the correlation between virus and different disease. Infectious disease is a big problem. I also want to put one focus on the bacteriums, the microbiomes projects there. If you look at all the human cells in the body, we are actually only 10%, right? So most of them are bacteria. Even you take shower 2 days, 2 hours a day, but still you are sort of covered by bacteria. Even you sort of wash your hand, after 1.5 hours, they all come back. So it's basically, I gave up to wash my hand anyway. But they are actually 300, if you look at the gene level, it's 300 times more. They cover everywhere. So it's not just, skin, nose, mouth, intestinal tracts, any places there. So they actually play it in a good way, right? They help you because they co-evolved with many, many years with your genome. So they actually help you digest the complex moleculars, produce vitamins, and doing this and that. But if they screwed up, they have developed all these metabolic disorders there. And actually not just immune disorders too. So we were the first group, together with the MetaHit Consortium, established the first gene catalog of the human intestinal tracts of the microbiomes there. It was 3 million genes, but now the updated version of catalog just be published in Nature Biotechnology. We sort of updated into 10 million genes, which is covered most of those abundant bacteria in the human individuals there. They grow from the infant, right? So, I mean, supposedly in the mom's body, so the kind of infant is kind of clean in their intestinal tracts there. But if you, after they're born, sort of born from the first week to the 1 year, so we did a comparison study together with Swedish group and actually sort of picked up 100 babies there and collected 3 data points with the mother's microbiomes there. And we discovered they really have a big shifting because of different, for example, C-section or natural born. Do they use formula feeding or actually breastfeeding there? Or do they take any antibiotics and, during the first year? So we actually had a very, very sophisticated analysis about all these microbiomes there. And we discovered lots of interesting things, just this few of them. For example, formula feeding. Formula feeding babies are more likely to require antibiotic treatment. Or actually, if you look at sort of different type of bacteria there, even the formula feeding and breastfeeding, they all grow bifidobacteria, but they are different bifidobacteria there. So if you have antibiotic treatment in the first years, they're actually most likely to develop a lot of autoimmune disorders in the future. So those kind of things have become very important. I don't have time to give the more details there, but when you grow sort of after 1 year of growth, they actually more likely to sort of with the adults population there. And if you compare with individuals, you find out the individual-specific enriched bacteria which are adapted to host the immune systems, viral infections, antibiotic treatments there. But for the common enriched, they are actually essential for survival. A lot of metabolisms there. Then we compare with, the sequencing of genome. Then we compare Chinese with Europeans there. Then we find out Danes drink a lot of wine and milk compared to Chinese, which is true. We drink a lot of Chinese hard liquor, which doesn't really show here. And also, Danes have a more gas-producing bacteria. That means they fart more, which is actually true. But we Chinese, if you look at our bacteria there, we actually have more antibiotic-resistant genes. That probably tells you we've been overused antibiotics, possibly. And also, we need a lot of essential, sort of enhanced essential amino acid and vitamin synthesizing bacteria there. That's just because of our diets are short of proteins in a way. But anyway, so those just for the common sort of ordinary people there. How about the disease? So we've studied all the bacteria and association with different disease there. Obesity is clearly very, very sort of linked to each other. So they have the biomodality for the low gene count and high gene count individuals there, which have less diverse bacteria and highly diverse bacteria there. And we know those low gene count group actually have higher fat percentage, more insulin resistant, so they're not really good. I mean, this is just one example. And type diabetes study. So we've done a Chinese type diabetes study there. 30 years ago in China, it's only less than 1% of the prevalence of the adults population. But now, it's almost sort of 1/4 of the Chinese adults either diabetic or to be diabetic. So it's really a big issue. And it's hard to believe your genetics changes in the last 20 years there. It must be something else. They say it's the diet's difference there, lifestyle, environment, or anything like that. So we did this sort of gut bacteria study. Then we discovered those, compared with genetics, they really have a very strong ROC curves using just the 6 bacteria we discovered in the type diabetes there. I list some of those known bacteria there. As you can see, most of those so-called beneficial bacteria are actually butyrate-producing bacteria, short-chain fatty acid-producing bacteria there. And they are actually helping a lot for the metabolism of the cofactors and vitamins there. And if you look at all those sort of harmful bacteria there, they are really oxidative stress resistance bacteria, sugar-related membrane transporter bacteria, whatever. So that just tells you from functional levels there, you also have quite different levels of them. But they also act as community, right? So you can see they are really sort of interact with each other. So you cannot just attack one problem, you have to think all of them as a community. So it's not just type diabetes, right? Autoimmune disorders. The rheumatoid arthritis, we actually did a Chinese study. We found out the associated genes there. The coronal heart disorders there, we also found out interesting bacteria there. Colon cancers too, even they have stage-specific bacteria or kind of markers we discovered. More and more. But how to manage the risk? I mean, we know those bacteria are not good, so how to manage the risk? Either by diets or by the different kind of pro or pre-diabetics there. The pro or pre-biotics there. But anyway, so what we've done there, we started to sort of get the Chinese traditional medicine book, which already by thousands of years experience, so they have to say, you know, they have whatever things are good for your metabolic syndromes there, and we test all of them. It's still ongoing, but this one sort of come on the top of the list, Aloe, which is pretty good. They sort of increase lots of shorty chain fatty acid bacteria there. I'm eating Aloe every day. Garlic powder is another one. That one my wife sort of does not really like. But they actually lower the bacteroides and also related with the bile acid secretion there, which is really promoted by the garlic powder there. Or probiotics, just adding some of those biotics there. So we are doing testing of Rosiberi bacteria, which we discovered is butyrate producing bacteria from our type 2 diabetes study. Then we use a mouse model there. They show very, very protective effects by even adding a lot of high-fat food there. So which just tells you potential in the future. Of course, people are talking about fecal transplantation, right? So it's really works. I mean, people not just testing the C. diff infections, but also type diabetes and things like that. But it's still a little bit disgusting. Even you put it into a dark chocolate, but still something we have to worry. So people in BGI started to really isolate and culture different bacteria from the intestinal tracts there, and started to make a cocktail or Poo Pills. But really, it's isolated cultured bacteria there. So we've already finished up, so it's not here. Well, we've already finished up more than 1,200 different strains of bacteria there. And we are sort of going to do more and to see in the future, based on your own metagenomics data, we could have a cocktail solution there. Well, eventually, the genome of itself is just kind of a box already programmed there. But how to really understand it, simulate it, change it, control it, it becomes a huge problem. Omics is the only way, right? To understand everything, I mean, by a systematic way, just to get all the layer of information, complete pictures there. So people are talking about data-driven for a long time, but data is not enough. I mean, we are only having a little data, but then we're complaining the analysis, how we could analyze those data. I mean, the only way to analyze data is to get more data. And the data sort of of course, people were sort of worrying about the cost of the genome. Right now, it's what, $1,000 US. If the trends continue, 5 years from now, it's $1 per genome. But when it is free, I want to get my genome free to be sequenced, it will come if the value of the data is much more than the cost of generating the data. I don't know how many of you want to sequence your genome at a cost of, let's say, $500. Do any of you want to sequence your genome at cost of $500? Oh, not many, huh? How about $100? Oh, more. How about free? Still somebody have hesitation there, which is good. That's real. But anyway, so it will come eventually. Everybody will be sequenced, probably for free. Some stupid company like BGI probably will offer that kind of options there, because we believe the value of the data is so much higher than generating the cost of generating the data. Well, that was one of the reasons we acquired a company called Complete Genomics in Silicon Valley in the Mountain View. So the idea, of course, is trying to generate data in a much efficient and cheap way. eventually, it's not just genomics, right? So you are reading all kinds of omics information there. And you've just seen these slides just on the first presentation there. But those kind of information has to be collected. And the future data generator will be in a single cell level, portable device, as you've already seen from the first presentation, real time and large scale. And eventually, those formula will be sort of drawn out. And by artificial intelligence, that would be my, I don't really believe human could really recognize themselves, so, or understand all this information by themselves using one single human brain. I would trust a computer more than a human brain. I know I probably offend some people here, but I feel artificial intelligence system like IBM Watson or whatever should be done and built up here. So with more and more data and training data sets and machine learning, that would be really important. At the time of when the computing power could surpass the human brain power there. But anyway, so we have to really learn how to live in the program the box, right? And we understand the box is changing because of somatic mutation. We should not cross the border. Sometimes we have to enlarge the box, manage the risk factors there by, for example, saving the necessaries. Can you save the life information just now? I mean, when you're still young, you save all the cells into refrigerators. And actually, those kind of information, digital information, will be there. Even you don't need to really sequence that now. But eventually, it will be used. I mean, when you're getting older, when the technology sort of bring it into another level, you could start to use that kind of information. Or you just heard 3D printing, you know, print a new tissue or anything like that would be very, very easy. Or reprogramming the life, right? So we know synthetic biology. We know the gene editing and all these issues there. I mean, people just cannot imagine how many ways you could try to change your already programmed life. But anyway, eat right, keep fit, live long, and when you need to die, die quick. This is really important, right? So you don't want to just stay in the bed for forever and never die. It's not just, actually, it's not just the emotional wishes there. It's not just, oh, okay. It just went through everything, okay. It's okay. So it's not just an emotional wish, right? So it's a rational choice there. What the final slide is actually I want to show, but it's already, clearly it's already displayed, is if the gene itself is selfish, right? So if you're programmed, whatever, if you have the power to change those programs already programmed in your life, I don't know if you could really manage those selfish genes. I don't know how that would lead the human individuals to what kind of, I mean, this is actually philosophical level, right? Questions there. It's not, should not be answered by scientists. But I worry, actually, if you really have the power to decode the life and change the programs, what they would lead us to. But anyway, science is science, so we will do that anyway. Leave all these problems to the others. Okay, thank you very much. And thank you for the listening.